Modern medical analysis often requires the use of precise liquid compounds, suspensions, and solutions. These media, typically combined in the laboratory at time of use, can be comprised of 2 liquids, a liquid and a powder, a diluent, and a lyophilized material. The resulting mixture is then dispensed either manually by droplet or sampled mechanically by machine or device. The process of accurately measuring, combining, and dispensing these media can be fraught with inaccuracies and error. Were the correct components combined? Were the correct volumes of the components used? Were the components mixed adequately? Was the mixture dispensed adequately? Bio-Techne's patented dual-chamber technology gives the user a convenient and easy-to-use device that is controlled, safe, and precise. The devices are each composed of 2 chambers, designed to hold materials separate and stable until time of use. The DCT-1 has 4 plastic components: the body, the inner chamber, the tip, and the cap. The body and inner chamber each contain a known volume of liquid, powder, or lyophilized material. When assembled, the inner chamber seats inside the body, followed by the tip and the cap. The tip assembly is designed with 2 functions. The bottom of the assembly has angled serrations to pierce the bottom of the inner chamber. The top of the assembly is fitted with a precise dropper tip for dispensing. The filled and assembled device can be labeled and packaged in any number of configurations. For end use, peel away the tear band on the cap. Screw the cap down, forcing the tip assembly serrations to shear the base of the inner chamber. Mix the previously separated media together. Unscrew the cap, invert the device, and dispense a droplet of the mixture as needed. The DCT-2 is similar to the DCT-1 in concept, but differs in materials and design. The DCT-2 can be comprised of 2 off-the-shelf vials or containers. These containers can be of different sizes and materials. Like the DCT-1, these containers each contain a known volume of liquid, powder, or lyophilized material. In this example, the lower chamber is a capped 4 milliliter glass vial filled with lyophilized material. The upper chamber, in this example a 2 milliliter plastic microtube filled with diluent, is sealed with our proprietary cap and collar assembly. Both assemblies can then be labeled and packaged in various configurations. When ready to combine the materials, the user takes one of the filled lower chambers and removes the cap. Next, the upper chamber screws onto the lower chamber via the cap and collar assembly. The user then peels away the tear band on the collar and presses down the top chamber into the lower chamber. This action causes the cap and collar to collapse into each other, shearing away the base of the cap. This allows the media from the upper chamber to mix with the media in the lower chamber. Once mixed, the upper chamber and cap and collar assembly are unscrewed from the lower chamber. The resulting compound, suspension, or solution is now available for extraction and use via instrument or manual device. Bio-Techne's 2-in-1 dual-chamber technology revolutionizes the ability to fill, store, combine, and deliver complex media in a simple-to-use, shelf-stable format. Best of all, the devices are customizable to satisfy both your and your end user's requirements. Talk to your Bio-Techne representative today about combining your technology with ours.